Hi, good afternoon, everyone. Maybe good morning for some. I would like to hand you over now to the moderator, Anna. Anna, can you please introduce yourself and also introduce the speaker? Anna is the moderator for this session today.
Hi everyone. It's a pleasure to have you here. Yes, here is still morning. I know that over there is already afternoon. This difference of time is always and sometimes a challenge for us to be able to work together. However, this is a pleasure to be here and to present Professor Luís Pereira de Almeida. My name is Ana Carvalho. I'm the head of Science Communication and marketing of GeneT, the Gene Therapy Center of Excellence in Portugal. And I'm also collaborating in the part of communication of Capacity 2023. So today we are here to speak about gene therapy, as usual in our in this series of webinars. And today we have with us Professor Luís Pereira de Almeida. Professor Luís Pereira de Almeida is professor, as associate professor at the Faculty of Pharmacy at the University of Coimbra in the center of Portugal. He also coordinates the Center for Neuroscience and Cell Biology and the Center of Innovative Biomedicine and Biotechnology in Coimbra. And also he's the coordinator of this brand new Gene Therapy Center of Excellence in Portugal. So he's very active in gene therapy since 1998. He has led more than 40 projects around the world with several and very diverse collaborations. He serves on scientific and strategic boards with research focus on brain diseases, namely in Machado-Joseph disease. Today, we have with us Professor Luís Pereira de Almeida. He will share with us some of his knowledge regarding the hope for rare diseases, the spotlight on case studies. So this is an amazing opportunity for us to have on this fifth gene therapy webinar series, this overview about some cases and case studies related with how can we treat and reach new treatments in rare diseases, and of course the opportunity for all of us to know a little bit more about GeneT and these amazing projects. So, Professor Luís, I think the floor is yours. Just to let you know that you have around 45 minutes, and then we will have some time for discussion and for all the amazing questions that I believe that our participants will have to give you after your presentation. So thank you so much for the opportunity to listen you and to share your knowledge with all of us. Go ahead. I think now the sound should be okay. So go ahead. Can you test it, please?
I can't hear anything. I'm fine. Can you hear me?
The sound is not amazing. I don't know if it's from my side or not.
I'm not sure whether I'm supposed to hear or not, and whether you can hear me or not. So I'll be waiting for your comment.
So people from the technical support, can you double check if you are getting good sound from Professor Luís, please?
No, we are not getting clear sound from it.
but we can hear you very well, Anna.
Okay, so but it's strange because usually it works very well. Luis, you are on the same computer, right? At least in me.
You can hear him, but this sounds a bit muffled. That's the check.
Anna, we can hear you very clearly.
Yeah, but not Professor Luis. So let's try to get a better sound from his side, maybe. Professor Luis, can you hear us? I think there is any trouble with his connection because I think he's not getting us. Let me call him. So I suggest Professor Luis to come out from the connection and get in again, and maybe we will get better sound from his side. Okay, so please, technical support, can you help me with this? I hope it works this time.
Why does it join you? Because you are very clear.
Yeah, we are not very close by. That was great if we could, but we are not. In the same city, but not in the same place.
Is he logging on?
Oh, he's probably
He's logged
getting...
off. Yeah.
So let's wait for him to return. So this is, I believe, an amazing opportunity for all of you to know a little bit more about Machado Joseph. That is the disease that Professor Luis and Holly's team are being dedicated since a while. So here in Coimbra and at University of Coimbra and at GNT, we dedicate ourselves more now. Now you can hear me? We can hear you much better,
Can
but
you
I
hear
think
me?
yes.
You can hear me, but I can't hear
Yes.
you.
But he can't hear
Okay.
us.
Let me check if the problem is with me.
But we can now the sound from his side is much better
Yes.
Okay, one
for me.
second.
Okay, so in case you cannot hear us,
MacBook Pro.
let's try one more minute. Sorry, everyone, for the
MacBook
ones that
Pro.
are waiting for listen, Professor Luis. But now his sound is much better. I hope
Should
that
be
he can
working
fix it.
okay. MacBook Pro.
In case he cannot listen us, we will tell him to go further, and then I will tell him. Now you can hear us?
Yes, yes.
Okay, great. Your sound is great also now, so we fix all the troubles. So
Okay.
thank you for these minutes, also for our participants. So I think now we are on time to know more about these all case studies that Professor Luis shared with us. So with the title Hope for Rare Diseases, Spotlight on Care Studies, now with us, Professor Luis Pereira de Almeida. Thank you.
Okay, thank you very much, Anna, for the kind introduction. So I will be sharing with you a little bit of history of gene therapy and some of the cases of success and others cases where things did not go well, but that also helped us to learn about the process. So I'm gonna try to share my presentation. Okay, let's see if this works. Can you see? Are you seeing?
Yes.
Okay. And so just to tell you a little bit where we are. So we are at CIBB, the Center for Innovative Biomedicine and Biotechnology, which is a large research center in the center of Portugal, with about 730 members and 320 PhD holders. And CIBB comprises Center for Neuroscience and Cell Biology, CNCUC, a center that was created almost 50 years ago, and ICBR, a center more linked to clinical and biomedical research. And it also includes two teaming projects. Teaming projects are large European Union funded project to create centers of excellence. In this case, we have MIA Portugal on aging and GeneT to create a center of excellence on gene therapy. And so this gives you a little bit of a flavor of where we are and what we do. We work on these different four areas, mainly neuroscience and disease, metabolism, aging and disease, innovative therapies, and healthcare challenges. And we are supported by very good research and innovation support units and scientific platforms with experts to help the researchers to provide the best research possible. Now, gene therapy, as you know, and I'm talking here to people that are already converted to the interest in the advantages of the gene therapy, but it brought some extraordinary successes during the last years, such as the one on spinal muscular atrophy that we'll talk a little bit in more detail later on, Zolgensma, and also the treatment of Leber congenital amaurosis, a disease that causes blindness, and that we also got fantastic results for this disorder. Now, gene therapy in Europe has a relatively narrow definition. It involves a class of medicines that work by inserting a recombinant DNA or RNA into the organism, and this is a definition that is much more narrow from the definition as compared to the definition of the Food and Drug Administration in United States. It might be revised in the next years because it is leaving out, for instance, the antisense oligonucleotides. Now, gene therapy is the corollary of 150 years of biomedical research and innovation, starting with Mendel's laws and Charles Darwin, the theory of evolution, and then with some of the hallmarks were the discovery of DNA structure by Rosalind Franklin, Watson, and Crick. The recombinant DNA technology was also critical to allow us to manipulate the DNA, to cut it, to glue it together, and then to express it in cells. And at the same time, more or less in the 70s, people started to realize that we could use nucleic acids to do therapies. Of course, the tools were still not there. And during the 80s, a lot of the tools to carry the DNA or the RNA into our cells were developed, particularly the viruses. Major innovations was, for instance, the cloning of the AAV by Jude Samulski, and others did similar work for other viruses. And finally, we arrived to the first clinical trial in 1990 by the hand of French Anderson, which was also successful in terms of showing that it was a safe procedure. Of course, we have about 3 billion nucleotides and 22,000 genes. And during the 90s, the Human Genome Project mapped all these genes, and people realized that each one of these could be used as a new therapy. But again, there was the question, what was the best system to deliver these genes? And people understood that we could use either viruses or non-viral vectors. Non-viral vectors are basically formulations of lipids or polymers with the nucleic acids. And in general, they are relatively safe, but they are relatively inefficient as compared to viral vectors. Viral vectors are basically viruses that have been modified to carry the DNA or the RNA. And they are much more efficient because they benefit from the evolution over millions of years that allows the viruses to be extremely efficient in terms of parasiting our cells and carrying the genetic material into our cells. And so most of the clinical trials have been made with viral vectors. These viral vectors basically involve the removal of all the genes that are involved in the pathogenicity of the original viruses, and then splitting the viral genome into at least two pieces of DNA. One where we have a packaging construct, where we have the genes that encode for the proteins that will assemble the viral particle. In the second plasmid, typically where we're going to have our gene of interest flanked by regions that will allow this transgene, as we call it, to be packaged into the viral particles. Now, when we put these two segments of DNA into cells with the help of a reagent like lipofectamine or others, we can basically the cells will take this DNA and use it as if it was their own DNA, and will be transcribing and translating proteins that will self-assemble and that will incorporate the DNA or the RNA into the viral particles. And these viral particles we can purify, concentrate, and whenever we need it, use it to carry the genetic material into our cells. Now, we can have basically two major approaches. One is the direct delivery. So we take the viruses and we inject the viruses directly in the organ or tissue that we want to treat. Or we can take cells from the patient, modify these cells with the vectors, and then put back the cells into the patient. And we call this ex vivo gene therapy, while the first we call it in vivo gene therapy. For ex vivo gene therapy, we typically use integrating viruses, such as retroviruses derived from the Molonian leukemia virus, or lentiviruses derived from, for instance, HIV-1. These viruses will integrate the genetic material into the chromosomes of the target cells. And so as these cells divide, they will preserve the transgene. They will transmit the transgene to the daughter cells. And so we will have a permanent expression of our gene of interest, not only in the cells that we modify, but all the cells resulting from the division of the original cells. And so this is why it is typically used for ex vivo gene therapy. For in vivo gene therapy, typically we use viruses that do not integrate the genetic material, such as AAV vectors. The AAV vectors, adeno-associated viral vectors, became the most popular viruses nowadays for in vivo gene therapy. Of course, adenoviruses are still used, particularly for cancer therapy, and herpes simplex derived viruses are less and less used, I would say, but have been quite widely used at the beginning of gene therapy. One of the advantages of gene therapy in general is that we can have a very prolonged expression of our therapy. And so this is just an example of at the time when I started working on gene therapy, by the end of the 90s, I injected viruses encoding for beta-galactosidase, a reported gene, in the brain of the rats. And then I stained the animals that were sacrificed 1 week after the injection, 3 months after the injections, or 1 year after a single injection. And you can see that even 1 year after a single injection, we have a very robust expression of our gene of interest. So it's a kind of a change of paradigm, because we can have a very prolonged or virtually even permanent therapy after one single intervention. Now, at the end of the 90s, when I did these experiments, we were very enthusiastic. Everyone was very excited, and we were hoping to be able to treat or even cure many diseases. But unfortunately, 2 accidents happened more or less in 1999 and in 2000. One resulted from an acute immune reaction to adenoviruses, and another one resulted from integration of the turn gene near an oncogene. And so basically it activated an oncogene and led to leukemia in some of the patients. And at least one patient died after this gene therapy. Also the same with the adenoviruses. One patient, Jess Gelsinger, died resulting from this acute immune reaction. And so this was terrible for these patients, of course, and their families. And the clinical translation of gene therapy was put on hold for some years. But of course, people explored other applications, and the technology also evolved. One of the applications that we developed was to use the viruses to produce models of disease. As you know, when we start to work on a new disorder, one of the most important things that people ask is there an animal model or is there a cell model that allows us to understand better the mechanism of the disease and to try new therapies. And so we were struggling with an animal model at the time, and we thought, why don't we use the viruses to express the mutant protein in the brain of the animals, and in this way to create a model of disease. And this is a strategy that at the time was very innovative, and that is now widely used in many labs around the world. Now, finally, after 1 decade, in 2012, uniQure was able to have its medicine Glybera approved to treat this disease associated with mutation in a lipase. And basically, these patients have trouble to process lipids, and they will accumulate these lipids, and they can develop pancreatitis, and this can be fatal. Now, the therapy involved basically AAV vectors encoding for this lipase that were injected in the legs of the patients, and it was effective. But it was not a commercial success, unfortunately, and the company was forced to stop the commercialization of the drug because there were no clients to buy it. But still, it opened the way for many other medicinal products involving gene therapy. One of these was Luxturna, that I already mentioned at the beginning. Basically, these are AAV vectors encoding for RPE65 that are injected close to the retina, and this prevents the kids from becoming blind. And so this was a huge success. Another huge success was Kymriah. Kymriah was the first CAR-T medicinal product. Here, people use lentiviral. This was work done by Novartis. They used lentiviral vectors to modify the T lymphocytes in such a way that they express this receptor that will allow the T lymphocytes to stay activated and to recognize the B lymphocytes that are overproliferating in leukemia, in lymphoblastic acute leukemia, and to destroy these overproliferating B lymphocytes. So this was a major success. Now, as you know, there are at least 6 products that are approved for commercialization involving this same principle of the CAR-T, and it was really a game changer, I would say. Another major success was the work of Brian Casper, that basically discovered, first of all, that AAV9 vectors are able to cross the blood-brain barrier when injected intravenously and to reach the brain parenchyma, at least in very young patients. And so the viruses cross the blood-brain barrier and are able, use it to put back the gene or the cDNA encoding for the survival motor neuron protein. And in the most severe form of this disease, patients die before the age of 2 without ever getting the capacity to control the movements of their arms or legs. And now you see here on the right a patient that was treated and can do a more or less normal life. So this was a major breakthrough that opened the way for many other approaches that involve gene therapy for brain disorders. Now, one of the things that also is interesting is that we are using, in fact, technologies that can be used also for vaccination. And in fact, the vaccines that we all took during the COVID-19 pandemic from AstraZeneca or from Pfizer or Moderna, in fact, are very much alike the gene therapy medicines that we take for other applications. So basically here we have an adenovirus. Here we have a messenger RNA in a lipid nanoparticle. In both cases, this was widely used in millions of patients without the major side effects. So a demonstration that gene therapy can be brought to a large number of patients at a low price and also without severe side effects. Now, gene therapy for hemophilia has also become a success, both for hemophilia type A and type B. And basically here we have AAV vectors encoding for factor 8 or factor 9, depending on the kind of hemophilia. These vectors are injected intravenously, they go to the liver, they transduce the liver, and the liver starts to produce these factors. And by doing this, patients can do a more or less normal life without the need for these weekly infusions of these factors. So also a very interesting success. Of course, as the liver cells divide, there is no guarantee that this will be a therapy that will last for the whole life of the patients, but still it is expected that it will be effective for a large number of years. A major success also was already obtained with gene editing. It proved to be effective in sickle cell disease and beta thalassemia. And so in the most simple form of gene editing, it basically allows the production of fetal hemoglobin, and the patients can do also a normal life after this therapy. Now, we have been working here in Coimbra in neurodegenerative disorders, and particularly a group of disorders that are the trinucleotide repeat diseases. The most well-known is probably Huntington's disease, where we have an overrepetition of a trinucleotide within the gene encoding for Huntingtin, and this transcribes and translates into an Huntingtin protein with an overlong polyglutamine tract that becomes toxic. But there are other similar disorders. One of these is SCA3 or spinocerebellar ataxia type 3. where we have the over-repetition of the trinucleotide CAG within the gene that encodes for ataxin 3. So what happens in this disease is that a mutant ataxin 3 that has an overlong polyglutamine tract will be generated, and this mutant ataxin 3 with the repetition of the glutamine amino acid will cause neuronal dysfunction in different regions of the brain, particularly those regions involved in control of in motor control. Also, a hallmark of these diseases are these aggregates of ataxin 3 that appear in the nucleus of the cells, and that have also been associated with the disease. So patients will develop this trouble to walk, to equilibrate, to articulate the speech. And so it is a progressive disorder that will develop and that will cause huge suffering to patients. We have been developing different approaches to address this disease. These involve autophagy inducers to promote the clearance of this mutated and aggregated protein, antisense oligonucleotides, calpain inhibitors, viral-based gene therapies, gene editing, cell transplantation, and I will mention a couple of these approaches that we have developed. And as I told you, one of the strategies that we developed very early was the use of vectors to produce models of disease. I did that first for Huntington's disease, but later we also did it for Marshall-Joseph disease or spinocerebellar ataxia type 3. It's the same. And here you see when we inject the vectors on the right hemisphere encoding for mutant ataxin 3, we see these aggregates of ataxin 3 with these ubiquitinated aggregates, nuclear inclusions, in the loss of the normal markers of this region of the brain. So if we look at the cell, and here is the nucleus, we see that the DNA with the mutation will be transcribed into the RNA. That will be translated into the mutant ataxin 3 that will be cleaved into fragments, and these fragments will have a tendency to aggregate and also to migrate to the nucleus where they further aggravate the disease. And one of the approaches that we thought we could develop was precisely to try to promote the removal of these aggregates of ataxin 3. Now, to do that, we relied on a strategy that will involve gene addition. That is one of the modalities of gene therapy. We have three main modalities: gene addition, gene silencing, and gene editing. And I will, show you an example of each one of these approaches. For gene addition, we knew that caloric restriction that had been studied in a laboratory that is close to ours was effective in stimulating autophagy. And so we submitted the animals to this caloric restriction, which has been shown to promote longevity, to promote neuroprotection, and to do this by activating autophagy. How is autophagy activated? It would seem that SIRTWIN1, a deacetylase, is activated, and when it is activated, it will remove acyl groups from LC3, that normally is a protein that is important in autophagy activation, but that when acetylated accumulates in the nucleus and becomes inactive. By doing the deacetylation, LC3 is able to translocate out of the nucleus into the cytoplasm and to activate autophagy. And what we did was precisely to submit animals to caloric restriction. One of the tests that we do is this rota rod. We put the animals here on the rota rod. We let them equilibrate, and when they are no longer able to equilibrate, they fall and they stop the timer. And you see that the non-treated animals are able to equilibrate here for 20 seconds or so, while the animals submitted to caloric restriction have a very drastic improvement in their motor performance. We confirmed this with other tests, and we also confirmed that when we submit the animals to caloric restriction, we have this decrease in the number of aggregates in this region of the brain, and the decrease also in the size of this lesion. But that beneficial effect is completely lost if we silence SIRTWIN1. Okay, so the number of aggregates goes up and the size of this lesion goes up, showing that the pathway through which caloric restriction is acting is through activation of SIRTWIN1. And so because of that, we decided to do this approach of gene addition, where we co-inject the vectors encoding for SIRTWIN1, and you see here the massive decrease from the control hemisphere into the treated hemisphere, the reduction in the number of aggregates, and the reduction in the size of this lesion. So this approach, this gene addition of SIRTWIN1 seems to be highly effective, as it is also the caloric restriction. So this was an example of gene addition. What about gene silencing? Well, because in fact, what we have is a gene that is mutated, that is transcribed into this mutant ataxin-3 and translated into the mutant protein. So if we promote the degradation of this messenger RNA, we should be able to prevent the production of the protein and prevent the disease. And this is exactly what we developed, an approach where we inject the viruses encoding for silencing sequences that will prevent the production of the protein. And you see here on the left the control hemisphere these aggregates of ataxin-3, it's one of these dots, and on the other hemisphere this massive decrease in the number of aggregates. Now, when we evaluate this also with an intravenous injection, we also saw some beneficial effects using DAV9. Now, another approach that we developed was the gene editing. This was developed in the group by Sarah Lopes. And so basically what she thought was we could use the CRISPR-Cas9 system to act not at the protein level, not at the messenger RNA level, but rather at the DNA level in the gene that is causing the disease. And so she developed vectors encoding for the Cas9, the Streptococcus pyogenes Cas9, in the guides, and vectors encoding for the guides that target ataxin-3. And she co-injected these vectors with a vector encoding for mutant ataxin-3 to induce the disease. And you see here in the control hemisphere the larger number of aggregates of ataxin-3, while on the other hemisphere where we co-injected the CRISPR-Cas9 system, a very dramatic decrease in the number of these aggregates. The same regarding this lesion caused by mutant ataxin-3 that was clearly decreased in the treated hemisphere. Now, this also is important that there was no massive, gliosis that we've seen with other systems when we use the CRISPR-Cas9 system, showing that it is well tolerated, and so that we can use this to promote the rescue of the disease. I didn't have the time to talk about the tail end constructs where our experience is different. So one of the aspects that is important is that, and that I already slightly approached, is this question of the routes of delivery, because it's not enough to be able to add a gene or to silence a gene or to edit a gene. You also have to do it in the sufficient number of cells, and particularly in the organ or tissue that you want to treat. Ideally, and in our case, we are dealing with the brain, we would like to avoid doing multiple injections in the brain that can have different issues regarding the surgical risk. And one of the approaches is to use this intravenous injection. This was work developed by Rui Nobre in my group, and the question he tried to answer was, can we deliver an allele-specific silencing sequences for Machado-Joseph disease, spinocerebellar ataxia type 3, by using intravenous injection? Of course, we are relying on the experiments that were done previously with for SMA, for spinal muscular atrophy, and the success of Zolgensma. And so we cloned into the AAV9 these silencing sequences in a microRNA backbone to reduce potential toxicity. And when we evaluated this, we saw that the treated animals do better in terms of motor performance as compared to the control animal that you see they're able to perform this. And the same with the rotarod that I had already showed before. And when we look at the brain, we also see these aggregates here, these little dots in red that are reduced in the treated animals, treated with an intravenous injection at postnatal day 1. And so this gene silencing approach by intravenous route seems to be working. Of course, it has some potential side effects because we are still injecting a very high dose of vectors. But we can discuss this at the end. These vectors are being improved to be more specific to the organ that we want to treat. Another approach that we evaluated was intranasal route. This was developed by David Rufino Ramos in my group. And the question he asked was, can we deliver these silencing sequences using extracellular vesicles? And so he coupled the silencing sequences to an exomotif to promote the enrichment of the extracellular vesicles with these silencing sequences. And also, he transduced these cells, these producing cells, with the DNA encoding for an RVG peptide, a peptide from the rabies virus that allows these vesicles to be better internalized into the brain. So he then administered these vesicles collected from the cells by the intranasal route in mice, and he saw that they were reaching the brain in the cerebellum, particularly these dots you see here in yellow. And also he generated a model of the disease that is very interesting. So basically, he generated lentiviral vectors encoding for mutant ataxin 3 fused to luciferase. So when we inject lentiviral vectors encoding for this ataxin 3 luciferase, we can detect the light in the animals using this equipment, the IVIS, and upon injection of luciferin, the substrate for the luciferase that is being expressed in the brain. Now, this allows us to monitor the expression of mutant ataxin 3, and when we co-inject the silencing sequences, we see the clear decrease in the light's signal. But what we wanted to know was, what about if we administer the silencing sequences by the intranasal route? And this is exactly what David did. He administered these vesicles to animals that had been previously injected with the vectors encoding for the mutant ataxin 3 fused to luciferase. And then he followed the light, and he saw a very clear decrease in the luciferase activity in the treated animals, the animals treated with the vesicles, and also a decrease in the mutant ataxin 3 messenger RNA. So this also seems promising. And finally, I would like to tell you a little bit about the approach of using the injection in the cerebral spinal fluid by injecting in the cisterna magna. So this was work done by Ana Carolina Silva. And basically, you have here the different approaches of getting a gene therapy medicine in the brain. You can use an intravenous injection, but this has the side effect that there will be a very large transaction of organs like the liver and the heart. You can use the ICV injection, the injection in the lateral ventricles. This will allow the transaction of a part of the brain, the frontal part of the brain mostly. Then we have lumbar puncture that is being used for Zolgensma. So you inject in the interthecal space, and this is particularly adequate if you want to treat the spinal cord. And then you have the ICM injection. So here there is this is cisterna magna where we can inject directly without the need for a craniotomy and for an injection in the parenchyma. And this is exactly what Ana Carolina tried to do. This should allow a good spinal cord transaction, brain transaction, and low toxicity for the dorsal root ganglia. So she started by evaluating this in newborn mice that were injected with AV9 in the cisterna magna, and she saw the reported gene was transducing quite well the cerebellum. And so then she developed this AV9 encoding for these two short terpenes that were injected in the cisterna magna and of newborn mice, and then the animals were evaluated regarding the motor behavior and also the neuropathology. What we saw was that the animals that were treated, particularly with the high dose, were taking less time to do the task of over the beam of crossing this beam in these 5, 8, and 11 weeks after the injection. Also, we saw that the levels of mutant ataxin 3 were decreased, and the GFAP levels were being decreased. So the gliosis was being decreased. And the NeuN levels, this neuronal marker, are being increased, and there is a dose response effect of the viruses. Moreover, we saw that there was a reduction in the number of these aggregates of ataxin 3 in the treated animals. So we also evaluated at the transcriptomic level the expression of different genes in the animals, and you see here in pink the wild type non-treated, in green the transgenic non-treated animals, and in blue, so in the middle between the wild type and the non-treated animals, the animals that have been treated with these vectors. So this seems promising. I didn't go through all the work that we've done with this, but we demonstrated that it is allele specific, decreasing the levels of mutant ataxin 3 without decreasing the levels of wild type ataxin 3, and that this is effective at the neuropathological level in another mouse model that I didn't have the time to show you, and also in this very severe mouse model that I showed you. So this seems closer to patients, and hopefully these different approaches that we have developed all seem promising, but particularly this approach of injecting in the cisterna magna this microRNA-based gene silencing is already quite advanced and close to the clinics. We are also monitoring patients to identify biomarkers and to establish a trial-ready cohort. I didn't have the time to go through this. A lot of people have been contributing to this work, and I would like to acknowledge all of them. And before I finish, I would just like to say a few words about GeneT, this gene therapy center of excellence that we are putting together. So we have been working for more than 25 years in the area of gene therapy, in some cases with companies, in some cases with academic funding. We have been running also an infrastructure that is part of our national roadmap of scientific infrastructures to produce and to make available viral vectors for the whole scientific community in Portugal and abroad. We have an ecosystem that is favorable in terms of having access to the different faculties, to a biotechnology park that is close by, to a clinical academic center, to a large hospital with 2,000 beds and a lot of experience in innovative procedures. And we have an unmet medical need because we have 7,000 rare disorders that affect 6% of the human population and in 95% of the cases without the therapy. So there is a need to establish centers of excellence on gene therapy. And for this, we established a partnership with the University of Sheffield and the University of Eastern Finland. Both universities have already gene therapy centers up and running for manufacturing particularly. And so we are building on this expertise from Mimmo Azus and from Seppo Ertuala on the manufacturing. And so we applied to funding from the European Commission that generally funded this gene therapy center of excellence that will allow us to establish a new building close to an existing biomedical research building with the capacity to manufacture and to do research and clinical trials. So the way we thought of this is that we will have a research and development unit that will allow us to work on other targets besides the brain that on which we have already a reasonable expertise, that will also allow us to expand our production unit and to manufacture it under GMP. And finally, a clinical trials unit to do small clinical trials, investigator-initiated, involving gene therapy, so that we complete the loop of translation and hopefully be more effective than until now. We have been also assembling more critical mass with other projects such as GCure that allows us to have the support of Jude Samulski on the manufacturing, GeneH to reinforce our capacity with other stakeholders, particularly patients, government, hospital, and the industry. Herdera, that is the largest initiative on rare disorders, where we co-lead the work package, the technology work package, to develop new platforms of gene therapy involving AAV vectors, RNA, gene editing, and lipid nanoparticles. We are also working on messenger RNA and creating the capacity with the Syntec project and on messenger RNA, and with the RDAT project on AAVs to solve still some of the issues that they have. The ESME project allows us to maintain a trial-ready cohort and to study it in order to establish the biomarkers, and the capacity has been allowing us to collaborate with you in Tanzania and Uganda, and hopefully to further develop this network of expertise as we did with another gene therapy center of excellence in Slovenia through this project, GeneH. These are some of the partners that have been collaborating with us, and we hope that we'll be able to overcome some of the limitations that still exist on gene therapy. In particularly, this Herdera project is expected to reinforce our capacity to address rare disorders. And with this, I would like to thank you for listening, and we'll be happy to answer any questions that you might have.
My camera is not working. Let me try to fix it. Thank you, Luís, for your. Let me maybe. I don't know what's happening. No, I did something on a meanwhile that is not allowing me to share my screen. So thank you, Luis, for your presentation. So I think now we are open to questions. So you can do it on the chat, or you can ask to put it your questions directly online. No questions by now. So any questions from your side?
Go ahead. We have already hands raised on ICT support and from Okelo, Okelo Duke. Go ahead.
Okay, this is Sisi from JCRC.
Hello,
Thanks,
Sisi.
Professor, for
How are
the
you?
very informative presentation, and exciting things are happening there, including opening the Center of Excellence, similar to what we are planning here, but not for rare diseases. So I have a question and a comment. One, you talked about the route of administration where you are doing IV routes for, I think it was silencing or one of those three. So how does the product get to the brain through the blood-brain barrier? So that's one question. The second is, there are other rare diseases now that have gene therapy products on the market, like the AADC and the CALD. And for the AADC, they put the product directly in the brain. I'm not sure about the CALD. What can you learn from those gene therapy products on the market already that cause brain issues? Yeah.
Okay, thank you very much, Sisi. So the crossing of the blood-brain barrier is still something that is under investigation. We know that, for instance, when we use serotypes like AAV2, basically the vector will transduce the endothelial cells and will stay there. So instead of crossing the endothelial cells and reach the parenchyma, it will, by transducing the endothelial cells, will stay there instead of continuing their way into the brain parenchyma to neurons. But it is still something that is under investigation. There are a lot of research on establishing new capsids, because the capsids that people have used until now are mostly capsids that exist naturally in nature. Okay, now the next generation will be completely different. People have been working on different approaches, either rationally modifying the capsid or just doing directed evolution of the capsids in order to produce new capsids, completely new, that are that will be much more effective in doing this job of crossing the blood-brain barrier and reaching the neurons or the astrocytes. And with even there, you can have selectivity regarding the cells that you would like to treat. And we can speak of the brain, but of course, this kind of work is being done for all the organs. Okay. And even for in some kinds of disorders, people are considering, for instance, to generate CAR-T by an in vivo approach. If we are able to generate a viral vector that reaches the T lymphocytes specifically without transducing the other cells, we could be able to inject intravenously the viruses, and they would generate the CAR T cells directly without the need to remove the cells from the patient expand, culture it, modify, and put it back, which is really a logistic nightmare. So in the future, I see more and more this capacity of selective targeting our target cells to become the whole and to become much more efficient. The other question you had was the question, there are some diseases indeed where people have been injecting directly the vectors in the brain parenchyma. It will depend a little bit on the turn gene, first of all, whether the turn gene or its product can have a bystander effect. So if, for instance, we're talking about a protein that can migrate from cell to cell, it is not as critical to transduce a large number of the cells of the brain that you would like to treat, because each one of the cells will kind of spread the protein, and by doing that will still be effective despite the low number of cells that are transduced when we do an intraparenchymal injection. Because the issue with the intraparenchymal injection is that the viruses only migrate a couple of millimeters. And so if you want to go beyond these couple of millimeters, at least for now, you'll have to do multiple injections. Or you would have to use a route of administration that allows you to reach all these cells. Did I answer your questions, Cici? Dr. Cici?
Yes, I think, yeah. So I think the technology is improving every day. And I think we can do more with what is coming up and addressing some of these questions, yes.
Absolutely, thank you.
Thank you. So we have a couple of more questions. So one is from Timothy Lumu. What really drives the cost of gene therapy models? What are the suggested subsidization interventions?
So the question of the price, if I understand correctly, yeah, there are multiple levels, okay, that to drive these high costs that we have. But there are a lot of different strategies that are being developed to reduce the costs. One is the platform approach. So if we develop, for instance, an AAV or a lentiviral vector that will be more or less the same independently of the turn gene that we use, and that we have an extensive characterization of knowledge about it, when we do the change of turn gene, hopefully a lot of the approvals and the demonstrations of safety that we typically have to do for the approval by the regulatory authorities will already have been done. And so this is a little bit the idea of the platform, to make it easier for the evaluation of the safety, to build on something that is already well established. And by doing that, this will facilitate the process of approval by the regulatory agencies. Now, this will also have other advantages, that is even the manufacturing. So the manufacturing, people are working on improving the capacity of manufacturing at lower prices. And there are a lot of different strategies to do that, like increasing the yield of the producing cells, reusing some of the materials, the consumable and plastics that nowadays are being thrown away, So there are a lot of different aspects that can be improved and on which people are working. Um, I think, examples like the production and the hospital exemption that we've seen being employed in Spain and other countries also, as you know, allowed the decrease of the costs from €500,000 to 50,000 euros. So it is possible to decrease to 1/10 of the costs. And I think we can even expect to decrease even further. And so these are things on which we are working to make the process cheaper. And also by putting everything together in the same spot, in the same institute, like we are trying to do with GeneT, we believe that we will facilitate the whole process and decrease the costs of production. Did I answer your question?
Hope so. Anything, I think Lumu can send us a message. So let's move to the next one. So Kenneth Mail, it seems gene therapy for rare diseases is taking a big step already. What could be driving this, and what could be limiting the acceleration of development of gene therapy for rather common diseases?
Yeah, I think the rare diseases have some issues, okay, but they have also some advantages. So it will be difficult in some cases to have a number of patients sufficient to reach the statistical significance. And so in many cases, things that for other disorders can be done, the trial in only one spot and one center, we'll have to go to multicentric to enable us to reach the number of patients that are necessary for statistical power. So this is a problem that non-rare disorders, more prevalent disorders, will not have, and so it will be easier to do trials in disorders that are more prevalent. I think that the rare disorders are like a good test tube for the more prevalent disorders. Typically, in rare disorders, you have a very clear genetic cause that can be addressed with a monogenetic therapy, while in more prevalent disorders this can happen, and when this happens it's easier, but we also have more prevalent disorders that are multifactorial, and these will be much more difficult to address. A very good knowledge of the mechanism is necessary, and also tools that allow us to do multi-gene approach instead of relying on a single gene. So I think this gives you a little bit of the perspective on this. I don't know if I fully answered this question, but yeah, I still think that the rare disorders are a kind of a good test tube for more prevalent disorders.
Now we have a couple of questions about sickle cell disease. One is asking, how do you feel about frequent mRNA injections for sickle cell disease treatment? And the other one is, after sickle cell treatment with the gene therapy, can the person pass on the genes to offspring? And in case of marrying a career or fellow warrior, can then end up with having warrior?
Well, no, the thing is, gene therapy is being done only in somatic cells. Okay, so there is an agreement at the global level not to intervene on the gametes on the germinative line. So basically, all the gene therapy approaches that we are developing in every country in the world are exclusively circumscribed to this patient. They will not be transmitted to the descendants. Okay, so there is no risk of transmitting if the things are done properly as they are being done. There is specific protocols to avoid the risk of transmitting to the descendancy. And there is an agreement between all the researchers to do it like that, at least until the technology to intervene on the germinative line is sufficiently safe, which is not the case at the moment.
There's also a question about what factors could potentially lead to gene therapy treatment to be unsuccessful.
Well, some of the factors we've already seen. Okay, so the immunogenicity in some cases of the vectors. For instance, in the AAV vectors, one of the issues is that a part of the population already has antibodies against the AAVs because the AAV is a very common virus, actually. One thing that I forgot to tell is that there is no disease associated with AAVs. And so most of us have already been infected with AAVs and did not develop any disorder. But also many of us have antibodies in sufficiently high levels to prevent treatment by AAV vectors. So how do we solve this? There are ways, okay? There are protocols of immune suppression that allow us to get rid of the antibodies during a transient time, during a short period of time, that gives us a window of time to inject the vectors and to perform a treatment, okay? So this is one of the problems, but other problems are there. For instance, if depending on the disease, we might need a silencing over 90% of the normal levels. This might be difficult to achieve, and also the number of cells. If we take a disease where you need a very high number, a very high percentage of the cells of the organ to be transduced, this can also be difficult to achieve. So there are a number of problems that still need to, there's plenty of work for students and researchers who want to embark on this area that is very promising, very exciting, but there's lots of technical problems still to solve.
Now we have Benjamin Bakeene asking about what we talk about CAR-T cells therapy, and he would like to know more about how do you use CAR-T on Machado-Joseph disease specifically, and secondly, why do you think that cell and gene therapy is lagging behind for infectious diseases compared with non-infectious diseases?
Mm-hmm. Well, for the CAR-T therapy, we haven't used for Machado-Joseph disease, okay, because it's a disease where, it's not the most appropriate, I would say, in terms of approach to use the CAR-T. So why do we think cell and gene therapy is lagging behind for infectious disease? Well, I'm not so sure that it is lagging behind. If you look, the technology of gene therapy was used for the vaccines for COVID. And so it didn't seem to be lagging behind, actually. So it did quite well. And I think in many cases in the future, we're going to see the messenger RNA vaccines because they are very flexible. Okay, it's relatively simple to design a new vaccine based on messenger RNA to just change the sequence and have a still effective vaccine for another target. And I think we're going to see it more and more. Now, for disorders like HIV, that it might be what you were thinking about. It still have some technical difficulties, but I think we're gonna get there. With the gene editing, it will be possible to inactivate the integrated proviruses of HIV if we are also able to reach the target cells in the in the bone marrow and the other in the niches where there are still cells that are infected. And I think it's just a question of time. People have been working on it, and we're gonna get there.
Okay, thank you. And we also have a question from one of our participants that put his hand up, but we don't know exactly which is the question. I don't know if technically we can give the words to Okello Duke. That is possible. Technical people, can you help me?
Hello, can you unmute and talk?
I couldn't hear.
Yeah. Okello, can you share your question with us? Maybe not. In the meanwhile, we have a question from Henry Odik. Can gene therapy be a good therapeutic approach in HIV, and how can this be approached?
I think I've answered already a little bit. I think so. There is a couple of problems, technical problems, but I think we're gonna get there.
Maybe I think we speak a little bit about this, but how do you feel about frequent mRNA injections for sickle cell disease treatment? Is a question from one of our participants too.
Well, it's theoretically feasible. But of course, wouldn't it be preferable for the patients to have something that you do a single shot, you'd have a permanent therapy? I think it would still be more interesting for the patients to have something that would produce a permanent effect. But the messenger RNA nowadays, people are working with the messenger RNA approaches to do gene editing, okay? And so we can envision a gene editing approach in vivo that would allow us to have a permanent effect. So in my view, that's, I think in the long run, that's the approach that will probably be used.
Okay, so any other questions? I think the majority has been already answered, the ones that you share with us. Anyone has any final question to Professor Luis? They are appearing. Let me check. So,
Yes, hello, can you hear me?
Yes. Go
It's
ahead.
Henry here. Yeah, it's Henry here at the JCRSC. Just a quick one for Professor. How long have they been working on the Joseph's disease? In terms of timelines, and also maybe at the moment where they with the product, they almost ready for the commercialization, anything in the pipeline, when are we likely to have it maybe out at that stage of commercialization, if there's any?
Thank you. So I'm not sure I fully understood, but we've been working on the approaches for Machado-Joseph disease for many years. But to be honest, at the beginning, we were more mechanistically interested, I would say. And also doing the translation process demands a certain learning process through which we went, okay? I think now we are much more equipped in terms of knowledge to do the process of translation, and I hope that this will happen, translation into patients within a much shorter time frame. But it needs the creation of infrastructure also that we didn't have at the time, and that we have been working on creating, and we're getting there. I'm not sure if this was the question.
Can you confirm, Henry?
Yeah, it's okay. Thank you so much.
Thank
Okay.
you.
Thank you. Any other questions? So we have a question from Apollo, asking about a shift on the support and on the where is coming the money regarding this shift focus away from mRNA research, and what you think about that.
Well, any disinvestment on research, in my view, it's risky. In my view, all the investments in research, or most of the investments in research, have paid off in the long run. And this has actually, there are many studies demonstrating profound effects of research at multiple levels, not only in terms of benefit from an health perspective, but also in terms of economical benefit for the population when there is an investment in research.
And I think we have maybe the final question that is maybe for the future. So the question is from Bayarugaba Yumar, asking, what are some rare diseases that are most likely to see curative gene therapy in the next 5-10 years? And what is the future of gene therapy?
Yeah, well, I was just a couple of weeks ago in a meeting to define a strategy for prioritization of the disorders that would first go into clinical trials, okay? Because there, of course, researchers developed a lot of different strategies. Many of these go to non-clinical studies in animals, and then the highest expenses are during the clinical trial stage. And so there are not enough resources to bring all the therapies simultaneously to clinical trials. And so there are some hard choices that might be needed to be done. Now, the way this study has been done is to have a number of questions. In this case, it was 27 questions that allows us to give a score, and based on that score, this will place the therapeutic approach for a specific disorder in a higher priority as compared to another. These different questions are aligned, grouped in 3 groups. So the first group is related to the knowledge about the mechanism of the disease. The second group on the psychosocial impacts over the patients. And the third group on the technology available to treat the patient. And all these are very important, but of course, if the technology is not mature enough, nothing can be, used. It's worthless if you know a lot about the disease, and if the disease has a very high psychosocial impact, without the proper technology to treat it, there's no point. But in some cases, you have the similar technologies, and you have to give a priority to diseases that afflict, children, for instance, instead of adults that progress faster, that you have a good knowledge. And in gene therapy, typically it has been easier to do gene addition. So you're missing a gene product, you just inject a viruses that encodes for this protein that you are missing. This is the easiest way, and most of the in vivo gene therapy medicines that exist are all of this kind. Gene silencing, it's a little bit more difficult, it will come, and gene editing can also be a little bit more difficult, particularly if you have a gene that you want to correct, a large piece of DNA that you want to modify. It becomes technically very difficult to do, and so I think gene editing in general, for in vivo use particularly, it's still not as advanced as gene addition or gene silencing, which are the modalities that are more advanced, at least for in vivo gene therapy. Of course, for ex vivo gene therapy, gene editing is already up and running, despite the low efficiency of it in general. I don't know, did I answer your question a little bit?
I think the question is answered. Any final notes, Professor Luis, that you want to share with us before we finish our webinar?
No, I think, just a message of hope. I think that the technology has been evolving a lot. The costs will go down, and I think it will be a fantastic tool that will alleviate a lot of suffering throughout the world. And so I think that your interest in this area is something that I think deserves it. Really, it's an area that deserves your interest. That's what I would like to tell. And I hope there will be more people working on gene therapy to solve the technical issues that still exist.
Yeah. So more people in order to reach more results in the early future, we believe. So thank you so much, Professor Luis, for your presence. And of course, I would like also to have the opportunity to be here with you. And thank you to all the participants that were with us during this 1 hour and a half. So it was a pleasure to share a little bit more about gene therapy in these our webinars. And of course, it's a pleasure to have you with us to know more about capacity and the opportunity to give further and get a better place for gene therapy in our society. So thank you so much. Have a good afternoon and see you around.
Same to you. Bye-bye.